So where do we get the fuel we need for exercise? Like other cells, muscle cells cannot directly use the energy released from breaking down glucose and triglycerides. Muscles need a specific form of energy for contraction. Body cells must first convert food energy or calories into ATP. The chemical bonds between phosphate in ATP and related molecules are high energy bonds, using the energy obtained from foodstuffs. Cells make ATP and its breakdown products, ADP, and a phosphate group. Conversely, to release energy from ATP, cells partially break down the compound into ADP and phosphorus. The released energy is used for many cellular functions. As soon as ATP is stored in muscle cells, it begins to be used. Another high-energy compound, phosphocreatine, is used to resupply ATP. An enzyme in the muscle cell is activated to split phosphocreatine into phosphate and creatine. This released energy can be used to reform ATP from its breakdown products. If no other source of energy for ATP resupplies were available, phosphocreatine would probably maintain maximal muscle contractions for about 10 seconds. The main advantage of phosphocreatine is that it can be activated instantly and can replenish ATP at rates fast enough to meet the energy needs of the fastest and most powerful actions. The disadvantage is that not much of it is made and stored in muscle. Carbohydrates are important fuel for muscles. The most useful form of carbohydrate fuel is the simple sugar glucose, available to all cells from the bloodstream. As you remember, glucose is stored as glycogen in the liver and muscle, and blood glucose is maintained by its breakdown. When oxygen supply in the muscle is limited, glucose is broken down into 3 carbon compounds called pyruvic acid. The pyruvic acid accumulates in the muscle and is converted into lactic acid. This produces 2 ATP per glucose and supplies about 5% of the total amount of ATP that could be formed from the complete breakdown of glucose under aerobic conditions. The advantage of this anaerobic glucose breakdown is that it replenishes ATP quickly, faster than phosphocreatine breakdown, and it provides most of the energy needed for events that require a quick burst of energy, or less than 2 minutes of work. The major disadvantages are that it cannot sustain ATP production for long periods of time, and that the rapid accumulation of lactic acid increases the acidity of muscles. If lots of oxygen is available in the muscle in aerobic conditions, the bulk of the 3-carbon pyruvic acid is shuttled to the mitochondria, where it is fully metabolized into carbon dioxide and water. This breakdown yields approximately 95% of the ATP made from complete glucose breakdown. Aerobic glucose breakdown supplies more ATP, but it releases energy more slowly. This slower rate of aerobic energy supply can be sustained for 2 to 3 hours. Here is a diagram that shows the ATP yield from both aerobic and anaerobic glucose use. Here you can see that anaerobic yields significantly less energy in the form of ATP than does aerobic. Glycogen is the temporary storage form of glucose in the liver and muscles. Muscle glycogen is only used by the muscles, and liver glycogen can be released into the bloodstream to increase blood sugar. During low to moderate physical activity, this can sustain work for up to 2 hours. Endurance athletes sometimes reach a point of extreme physical and mental fatigue, called hitting the wall or bonking. This occurs because muscle glycogen has been depleted and blood glucose has begun to decline during exercise, leading to a deterioration of physical and mental function. Intake of energy is important in maintaining blood glucose during strenuous activities that last longer than 20 to 30 minutes. For these strenuous activities, it is recommended that individuals intake 30 to 60 grams of carbohydrate per hour, which can delay fatigue by 30 to 60 minutes. When fat stores in body tissue begin to be broken down for energy, triglycerides first yield 3 fatty acids and a glycerol. The majority of the stored energy is found in fatty acids. During activity, fatty acids are released from various adipose tissue deposits, the largest storage form of energy in the body, into the bloodstream and travel to the muscles, where they are taken into each cell and broken down aerobically to carbon dioxide and water. Trained muscle cells have more mitochondria and a greater ability to use fat as fuel. For each turn of the citric acid cycle in the breakdown of fatty acids, we yield 12 ATP, meaning that a 16-carbon fatty acid chain would yield 108 ATP for energy. Fat has the greatest potential for energy release. As exercise increases, the rate of fat usage can increase, but it is dependent on the concentration of fatty acids in the bloodstream. In intense physical activity, fat is not a major fuel source because it requires more oxygen for breakdown than does glucose. However, in prolonged exercise, using trained muscles, stored fat can be a preferred energy. Although amino acids derived from proteins can be used for fuel in muscles, their contributions are relatively small, compared to that of carbohydrate or fat. Most protein is reserved for building muscles and replenishing body tissues, as well as important for making enzymes and hormones. As a rough guide, protein only provides about 5% of the body's energy. During endurance exercise, protein can be contributing up to 10 to 15% of needs, especially as glycogen stores in the muscle are exhausted. Most energy supplied from protein comes from the breakdown of branched-chain amino acids, and a normal diet provides ample amounts of these amino acids, so supplementation is not required. Here you can see that different types of energy sources are used for different types of exercise. In most activities, carbohydrate is the main source of fuel. In weightlifting, fat can provide additional fuel sources, and in endurance sports, fat and protein both have an increased contribution. Here you can see the different types of energy sources used and the preferred by cells during different types of activities.